Hello and welcome to the second part of the hERG blockers video series. In this second part of the video series, we are going to do a very quick recapitulation of the first part of the video series, and then we will jump to the strategies and try to answer the question, how to design compounds aiming to avoid blocking the hERG channel. So, first things first. If you haven't saw the first video regarding the hERG blockers and the first video of the series, I would strongly recommend you to pause this video, go back to the first one, watch it from beginning to the end, and then go back here. It will make your understanding of what is going to be discussed here much, much easier. As a quick recap, the hERG is the human iter-agogo related gene. It's responsible for encoding a potassium-dependent voltage channel, and blocking this channel leads to what we call the long QT syndrome that can lead to ventricular arrhythmia and possibly the death of the patient. Several drugs are known to block this channel, and because of that, testing any clinical candidates for hERG is mandatory. You cannot test any drug in humans without proving that the drug is not a hERG blocker. The channel itself presents a large cavity, and because of that, it has a low selectivity, meaning that several different molecular frameworks can bind to this channel. And because of that, we have several binding modes and pharmacophoric models published in the literature. Again, we discussed that in more details in the part 1 of the video. There are several pharmacophoric models published for hERG channel blockers. Again, this is discussed with much more detail in the first part of the videos. Again, go back and watch the first part of the video if you did not yet. So most of the models, they describe the same situation. We have basic groups surrounded by hydrophobic units. The non-hERG blockers, in their majority, they present a basic group that is not essential, but most of them are basic molecules with hydrophobic, mostly aromatic groups surrounding it. So that combination of multiple binding modes and several pharmacophoric models and a large cavity, they lead to a high tolerance to structural diversity when you are trying to deal with the hERG channel. So how to avoid that situation? We can change physical-chemical properties by changing alkyl side chain sizes, inserting polar atoms, increasing the polar surface area, inserting negative charges or decreasing basicity, and also we can change stereoelectronic features, such as removing aromatic rings, increasing the number of sp3 carbons in the structure, also trying to induce some rigidification, and also changing relative positioning of functional groups in the molecule. One of the strategies we can apply is to try to decrease the alkyl chain size of a molecule that is a hERG blocker. Look at this molecule, for example, if we try to fit one of the pharmacophoric models to that molecule, we have the best fit would be the basic unit surrounded by hydrophobic groups here in green. One of them is an alkyl chain. If we can reduce the size of this alkyl chain or remove it altogether, in the end, we are diminishing or eliminating one of those hydrophobic binding units. Therefore, we can try to reduce the hERG inhibitory activity of this molecule. And this is what they did in this example. They replaced the ethyl group for a methyl group. It's a small reduction, but that made the trick. The potency of the drug stayed nearly the same, but the hERG inhibitory activity decreased from 12 micromolars to more than 100 micromolars. Also, we can insert polar groups to a structure. Again, if you look at those two molecules, again, we can fit both of them to the same model. Again, a basic group surrounded by hydrophobic units. The same thing for the second one. If we can insert a polar group in one of those hydrophobic units, in the end, what we cause disruption of that hydrophobic binding region. Therefore, we can try to reduce the ability or the capability of this molecule to bind to the hERG channel. So, in the first example, they inserted an alcohol in that tetrahydropyran unit in the molecule. The potency stayed nearly the same, but the hERG inhibitory activity decreased a lot, from 0.12 to 8 micromolars. In the second case, they inserted a phenol in the aromatic ring, also reducing the hydrophobicity of this region. So again, the potency improved a little bit, but the hERG inhibitor activity decreased from 3.7 to 22 micromolars. Also, we can try to increase the polar surface area. In other words, to try to increase the area where we have more hydrophilic groups in the molecule. That also reduces the ability of those molecules to bind to the hERG channel. If we try to, how can I say, to fit those pharmacophoric models to those molecules, the first one would be probably the basic unit surrounded by hydrophobic groups. The second one would be the basic unit surrounded by aromatic rings. Again, also, those are hydrophobic units as well. In the first case, they replaced the trifluoromethyl unit by a sulfone, increasing the polar surface area. So the potency of the molecule stayed nearly the same, and the hERG inhibitor activity decreased tenfold, from 3.8 to 31 micromolar. In the second case, they inserted a sulfonamide unit near to that bicyclic unit over here. So in this case, they had a good result. The potency increased a lot, from 250 to 30 nanomolars. And also the hERG inhibitor activity decreased from 23 to more than 100 micromolars. Also, one thing that we can do is to insert a negative charge in a molecule, especially if the molecule has a basic unit, making the molecule a deuterion. Well, why does that would work? We can try to fit those two molecules to the pharmacophoric model where we have a basic group surrounded by aromatic units. Remember that this basic group here is probably doing either a cation pi or an ion dipole interaction with some amino acid residues in the hERG channel cavity. If we insert a negative charge in this molecule, that ability of doing cation pi or ion dipole interactions with a positive charge will be greatly diminished. So in the first case, they just hydrolyzed the ester, freeing the carboxylic acid. The potency of the molecule stayed the same, and the hERG activity was greatly reduced. In the second case, that was a prescription drug. That was terfenadine. That was an anti-allergic drug that was withdrawn from the market because it was a very potent hERG blocker. And the insertion of this carboxylic acid at one of the edges of the molecule led to the discovery of fexofenadine. That is a prescription drug, also indicated for as an anti-allergic. The potency as an anti-allergic stayed in tolerable levels, and the hERG inhibitory activity decreased a lot, from 0.05 micromolars to 65 micromolars. One thing we must be cautious about that, when we have a drug that is able to exist as a deuterion, if the molecule can be permanently charged, that can lead to some absorption problems, because if the molecule is permanently charged, either positively or negatively, the drug will be much less likely able to cross membranes and much less likely to be absorbed. So that is not a strategy that should be applied without a lot of caution and prior thinking. Also, one thing we can do is to try to decrease the basicity of the drug. So if we try to fit those two structures to the pharmacophoric models, again, we have the basic center surrounded by hydrophobic moieties. If we can try to reduce that basicity, we reduce the ability of this positive center to perform or to be able to do either cation pi or ion dipole interactions with some amino acid residues in the hERG cavity. Again, if you are not remembering or not getting what I'm talking about, go back to the first part of the video series. So in the first example, they introduced a fluorine to the ring in order to reduce the basicity of the center. We can see that by the pKa, that reduces from 8 to 7. The potency of the drug stays on same levels, and the hERG inhibitory activity decreases from 26 to more than 100 micromolars. In the second case, also they introduce an alcohol to reduce the basicity of this amine here. We can see again that because of the pKa that is reducing from 9 to 8, the potency of the drug stays on same levels, and the hERG inhibitory activity decreases from 2 to 40 micromolars. So in the end, this is a very good strategy also to apply. Also, one other thing we can do is try to remove one aromatic ring out of the structure. So again, if we try to fit this molecule to one of the pharmacophoric models, we have the basic center surrounded by aromatic rings. If we eliminate or replace that ring for something else that is not aromatic, we can disrupt that pharmacophoric model a lot. In this case, they removed the benzene unit and left only a methyl group. And in this case, it was good on two senses because the potency for the desired activity increased a lot. And also, the molecule was a very potent hERG blocker, 9.5 nanomolars of IC50, and that activity increased from 0.0095, decreased to 7.4 micromolars. Also, we can try to make the molecule less flat by increasing the fraction of sp3 carbons in the structure. So again, this molecule would fit very well the basic center surrounded by aromatic moieties. And what they try to do is they replaced this aromatic ring, the pyridine ring, by a tetrahydrofuran in the second structure. Although it lost some potency, also the hERG inhibitory activity was also nearly abolished. So this is also a strategy to think about. One other thing that we can do is to try to make a rigidification of the structure. In other words, we can try to lock the molecule in a conformation that is not the conformation in which the molecule binds to the hERG channel. Sometimes this can work. So that molecule over here, again, would fit the basic center surrounded by aromatic rings. And what they did, they added another cycle to the structure, doing a cyclization of this basic center. So the potency, desired potency for the drug stayed the same, and the hERG inhibitory activity decreased a little bit. But again, that strategy was not as successful as one would say, because still 27 nanomolars is still a very potent hERG inhibitory activity. So probably more modifications would be needed to this structure in order to free that molecule from any kind of hERG inhibitory properties. Also, one thing that we can do is we can also do a isomerization, changing the relative position of functional groups in the molecule. This molecule over here can fit the model of the basic center surrounded by aromatic rings. And in this case, what they did, they changed the carboxylic acid position from the meta to the ortho position. The potency of the drug stayed the same, and the hERG inhibitor activity decreased from 50 to 126 micromolar. Of course, in this case, the isomerization has another effect. By pushing this carboxylic acid to the ortho position of the ring, that can also be twisting this ring out of the plane or out of position, also making it probably not being able to bind to the hERG receptor as well. So, wrapping up, the hERG is a potassium channel that plays a crucial role in regulating the heart physiology. Blocking this channel, it's an undesired effect. So because of that, we have to test any preclinical candidates for hERG blocking activity. It's mandatory. That channel is a problem because it has a low substrate specificity, meaning that we have different molecular frameworks, different binding modes, different pharmacophoric models of different kinds of molecules that can bind to the channel. So designing drugs away from the channel while keeping the desired activity intact can be a challenge, but it can be done. And the most common strategies, they circle around reducing the lipophilicity of the drug, reducing the flatness of the structure, and also reducing the basicity of the basic center of the molecule, if the molecule has one. So thank you for your attention. I hope you have enjoyed this video. Stay tuned for more videos from this channel.